So did you guys read the 2 truths and a lie? Do you know which one's true, which one's a lie? You ready to see Kanye? I ain't lying. Is it not true? You could even switch the labels, cross-section of the penis. Okay, so let me point out a couple things to you guys before we get into the lecture material today. First off, this is the week that you are going to feel overwhelmed for the first real time. You are starting the skull this week, the axial skeleton. There are a lot of people who run away in tears. There are usually tears the first week. I know a few of you guys have. How many of you guys have been to lab yet? Just a few. Okay. So it's pretty overwhelming. It seems relatively arbitrary that we make you memorize all those bumps and holes, but I will tell you that later on you will figure out that you learn the jugular foramen because the internal jugular vein goes through the jugular foramen, which is something you also have to learn. You learn the deltoid tuberosity because that's where the deltoid is going to attach to the humerus. So at this point, it's hard to get that kind of appreciation for what you're going through. So first, I empathize. It is difficult. Second, you are not going to do well on this stuff unless you study. It doesn't matter how smart you are. There is no way in hell you can learn this stuff without repetition. So let me give you some of the resources that we have available for you. Of course, you guys know that Open Lab is here. This is the week in which people will really, really start going to Open Lab. Okay, there are tons of times available for you for Open Labs. Let me point out a couple of other things for you. First, if you click on the resources page right here, I just put this up just a second ago. My lab aides, I run the lab aides, will lead peer study groups. This is the first one that's scheduled every week. They will be in the Redwood STEM Center to help you, not just with lecture, lab, but also with lecture. So 2:30 to 4:30 in the Redwood STEM Center if you want to go there. And I think it's right after Open Lab, if I remember correctly. Yeah. Lab ends at 1, so you can go over there. So it's not conflicting with Open Lab. And I think Open Lab is over here, isn't it? I can't remember. So there might be one on this campus, but that's up to their schedule. So I let them, hopefully we'll have one on this campus at some point too. So those are the lab aides. Be nice to them. If you're ever mean to them, they are my students. I will protect them, even if it's taking you down in the process, okay? You are my students too, but I want you to be kind and appreciative of one another. We have had people who really, really abuse them, and I do not appreciate that whatsoever. So in addition, there is tutoring available. The only time I know is once a week on the STEM Center, and this is one of the lab instructors that actually does tutoring. It's free. That's like separate than, like, she's actually an employee of the college. And then let me show you some other stuff that's available for you guys. So on the resources page, if you go to study tools right here, okay. So there's a bunch of stuff, but among them, a lot of you guys already found the printable flashcards. That's not the only thing though. There's also histology flashcards, and this web page I'm going to use today. There's a few web pages that are really good for histology that you can print stuff off of. So there's a bunch of resources for you, and they're all available for you for free. But one other thing is that the STEM Center on Redwood campus, we bought $30,000 worth of models, including skeletons, last year. And so that has all of that stuff for you to pull out in the STEM Center and look at. And this library right there also has skeletal system. So you can and should pull those out and repeat and repeat and repeat and repeat. That's the way to get this material into your mind. One other thing that you may or may not know, let me show you that this is here. There's tons of stuff available for you. It's on the restricted content page with the cadaver pictures, okay? And hopefully you guys have gotten the password for this. Yeah, they should have given it to you in lab. I can give it to you today, but I need to. Okay, so the password is for the restricted content is all lowercase, no spaces. Let me show you what's there, okay? These are not just pictures of any skulls. These are pictures of the skulls, for instance, that you have in lab. So these are pictures that you can look at. There are also down here, fill in the blank things that you can print out. And so you can do, I didn't mean to go to appendicular muscles. How did I get there? Fill in the blank. There's skeletal system ones. There's one for all different axial muscles, nervous system. So here's skeletal anterior view of the skull. So this would be great to, for instance, put in a plastic sleeve and do that stuff, right? So all of these resources are here for you. These were all made by my past lab aides for you guys to use. So there's tons of resources for you guys. So note to self, all of these print really easily, the ones without labels. When you get to these, they're not supposed to print easy if they are cadaver pictures that are labeled, because we wanted it to be hard for you guys to actually carry those around when they were actually somebody's grandpa. So the ones that are easy for you to print are really anonymized, right? So now if you're good at computers, you can print any damn thing you want off the internet, and you know that, right? Okay, so everybody cool? So there's lots of resources for you. Go ahead. No idea. I think it's just on the thing. I don't know. I don't think they're open on Sundays, but they are open on Saturdays. And the other campus is open on Saturdays too. Okay, you guys know where the STEM Center is on the Redwood campus? It's on the first floor of the SI building, the same building that has the lab in it. It's on the first floor though. Okay. All right, anybody have any questions? Take a deep breath and realize that this week is going to be difficult and challenging, and you can totally do it because people do it every damn semester, and obviously I did it at one point, right? I also remember how incredibly stressful and unpleasant it is, but I absolutely know that you can get through it. Okay, so let us keep going with histology. We're going to finish epithelial tissue today, and we're going to start connective tissue. Next week, we will catch up with lab when they're finishing bones. Well, when you guys are almost finished with bones, we'll catch up with bones. FYI, you don't learn the skull by me lecturing to you anyway. Is there any way in hell that I could actually teach you all the holes by projecting them up here? That's not how you learn the skull. What I teach you is the big picture understanding that helps you put together all of that memorization so that you can retain it, right? So lab is where you learn all the little holes because you actually need to have the 3D thing in front of you, okay? All right, so I don't remember last time, I think we were talking about polarity and basement membrane. Is that what we're doing? Polarity and basement? We just finished this page? Okay, and we talked about hemidesmosomes in here? Okay, cool. All right, so that means that very easily on a simplified picture like this, right, you should be able to A, recognize that it's an epithelial tissue, yeah? B, tell apical surface from basal surface, yeah? Basement membrane you could label, right? And even though I didn't ask you, you could also tell me what kind of tissue you would expect that to be. What tissue would be right here? Is B connective tissue, right? Not specifically yet. A, it's not drawn specifically enough for you to identify, but B, we haven't learned them yet, but you know it's a connective tissue that's going to be under an epithelial tissue. Good? Okay, so now, what we are going to do today is go through all the simple tissues and then go through all the stratified tissues. This is stuff that you've done in lab already, but in lecture you always learn more wise, more put it together into a story that hangs together instead of rote memorization. So I'm far more likely to ask you why would a tissue lining the lumen of the intestine be a simple tissue versus a stratified tissue in lecture than they would in lab? So you start thinking about functionality. You start thinking about why the tissue is structured the way that it is instead of just that's simple cuboidal epithelium, that's stratified squamous epithelium, right? So you're going to hear those questions as I start talking to you today, and your tests, for instance, will be a mix. It'll be a mix of identify this tissue and let's talk about the functionality of this tissue and why this tissue would be found in this location and this tissue would be found in a different location. Does that make sense? So listen to that too. Okay, so let's first start with the fact that, yeah, I think it's already been recording for a while. Yep. Okay, so let's start out with the fact that epithelial tissues have a naming convention that's relatively consistent. I mean, it doesn't work every time, but it works most of the time. And it is the three-name naming convention, and the three-name naming convention is adjective, adjective, noun, okay? And The noun is always epithelium. Okay? So you're going to have blank, blank epithelium most of the time. There are a couple of exceptions, right? Does that make sense? Now, note to self, if you leave off the epithelium, I will count the whole damn thing wrong. Because simple squamous is not a thing. Simple squamous what? Kumquat? Elephant? I have no idea, right? So you have to have epithelium, okay? So the adjective is generally, with a couple of exceptions. This one is how many cells you have from the apical surface to the basement membrane. How many layers, right? So this is going to be referring to the number of layers. And of course, the layer possibilities are, what do you call it when it's only one layer cells? Simple. What do you call it when it's anything more than one? Stratified. What do you call it when it looks like it's more than one, but it isn't? Pseudo stratified, okay? So we'll go through all of that today, okay? So, and then this one is the shape of an individual cell. And specifically, it generally is the shape of an individual cell at the apical surface. So this is the shape of an individual cell. And so these are the possibilities. If it looks like a fried egg from the side, it's usually even flatter than that's drawn up there, then you call it squamous, right? If it looks like an ice cube, right, about as wide as it is tall, then you call it cuboidal. If it looks like a column, you call it columnar, right? Okay. And generally speaking, and there are exceptions, squamous usually has a really flat nucleus this way. This one usually has a nice round nucleus, and this one usually has an oval nucleus, right? So if you're ever kind of in between, and you're like, I can't quite tell whether it's columnar or cuboidal, use that second check mechanism to see if that will help you, right? Does it do that every time? No, because nothing in biology works every damn time, but it helps a lot of the time, right? Helps a lot of the time. Good? Okay. So when we do epithelial tissue, we generally use the three-name naming convention, and there are exceptions, and we'll talk about the exceptions in just a second. Now, every once in a while, you're going to need one more adjective, like for instance, if it's ciliated, you put here, and then sometimes they don't use two. Sometimes it's like transitional epithelium. You're like, hey, I just taught you the rule, and now I'm not going to use it anymore. So, all right, so let's do the simple tissues. So first, let's talk about functionality of simple versus stratified tissue. Let's think about the extremes, okay? What do you think about a simple tissue versus a stratified tissue? Which one is more likely to be useful for instance, protection? Stratified, yeah, right? Okay. Now, as it gets thicker and thicker and thicker, it gets better and better and better at protection. But is it going to be really, really good at transport, bringing things in and out across the tissue? No. Simple tissues tend to be better at transport. Stratified tissues tend to be better at protection, right? Does that make sense? Okay, so that's sort of the trend that you see. Now, if it's only 2 layers, it's not dramatically thick, right? But if it's 25, you can say, oh, we're probably not trying to transport something across that very frequently. Does that make sense? Okay, so the simple tissues, quite often you will see that have something to do with transport. So let's look at them. So a simple squamous epithelium you are actually seeing right here, okay? A simple squamous epithelium, you should be able to look at it. You should be able to tell epithelial tissues because they're always going to have that polarity, that free surface and then a surface that's stuck to other stuff, yes? Okay, so you should be able to find. This actually has 2 epithelial tissues on it. We're only interested right now in the one at the top. It is simple because how many layers of nuclei do you see from the surface to the basement membrane? Just 1, okay? And it is squamous because the cells are squished flat. Yeah? Okay, so it's a simple squamous epithelium. This is going to be the absolute worst tissue in the entire body at protection. It's super duper thin. You like want that on the surface of your skin? Not unless you want your nose to bleed every single time you pull out a nose hair and all come out, right? So this is really, really good at transport and really, really abysmally bad at protection. So you find it in areas which are really set up for transport, like the alveoli of your lungs, where you do gas exchange, yeah? Or every single capillary in your body is made of simple squamous epithelium because they really want to get nutrients and waste products in and out, and oxygen, carbon dioxide in and out. Does that make sense? So this is a simple squamous epithelium. Now, In lab, they have a picture of simple squamous epithelium that's a top view. I will never ask you that picture because that's memorization. Because if you look at a top view, of course, it looks like a whole bunch of eggs next to each other, right? But if you look at a top view, that's just memorization because you can't tell that it's simple or squamous from that view, can you? No, right? You're just looking at a top view and it's like, hey, there's some cells, right? So they can ask it from you that way in lab, but I never will. I will never ask you to identify it from there. I will ask you, for instance, to identify it from here. So can you find the simple squamous epithelium in this picture? Yeah, it's right here. So you find the free surface, right? The tissue right next to it should be an epithelium. Check to see if it's simple and squamous, and it is. Yeah? So that's a simple squamous epithelium. Okay? Everybody good? Okay. So this slide has two tissues on it. This is also an epithelium, yeah? But what's this one? That's simple cuboidal, which is the next tissue we're going to do anyway. So let's talk about that. The next tissue that we are going to do is simple cuboidal epithelium. And simple cuboidal epithelium, first, let's loop back to what we did last week and say, what kind of section is this of this tube? Simple cuboidal epithelium. This is a section of a tube. What kind of section are you looking at? Longitudinal section. Yes? Longitudinal section of a tube. You get two parallel lines. Okay. What's this space called? Lumen. Okay, good. Now, of course, you can identify the epithelial tissue right next to the space, right? Okay. So this is going to be your epithelial tissue, and then all you have to do is go through the process of confirming that you can tell. It's both simple, because one layer of cells, as indicated by one layer of nuclei, right? And it's cuboidal because the cells are about as wide as they are tall. And what did I tell you about the nuclei? Usually nice and round. Yeah, that's a simple cuboidal epithelium, right? It's usually really easy to identify. I think it's kind of a cute tissue. I don't know why. It just looks kind of happy. Here's another one. Same tissue, different type of section. Can you find it? Yeah, simple cuboidal epithelium. Everybody good? Okay. This tissue is going to be really good at transport. You find it lining your kidney tubules. And I know you don't know what the hell a kidney tubule is, but let me just give you a preview of things to come. In your kidneys, basically you have miles of these teeny tiny tubes that are going to allow for exchange between your blood and your urine to make sure you get rid of the stuff you wanted to and hold on to the stuff that you didn't want to get rid of. Make sense? And it happens in these tubules. Okay? So they're really thin and there's lots and lots of surface area for this exchange. So lining kidney tubules. Go ahead.
So would that be cut like a transverse section?
Good, transverse section. Maybe a little oblique.
Yeah.
I can't tell whether the slide is just stretched or it was actually oblique, but it is, yeah. It's probably tried to be a transverse section, yeah, at the very least. Does that make sense? So it's the same histology, it's the same tissue, right? But this is a different type of section of the same tissue. Everybody good? Okay, so this is simple cuboidal epithelium. Good. And then the next one is the other simple tissue. There are two different kinds of them. And it's simple columnar epithelium, but I want to look at what the similarities of all of the simple columnar epithelia before I look at the differences. Okay? So what I should see in all of the simple columnar epithelia is one layer of cells from the apical surface to the basement membrane, right? Indicated by one layer of nuclei. Yes? And some people get confused because they're like, what if I see a nucleus back there? I don't care about that. That's not the layering that I'm talking about. I'm talking about making a 9-layer dip, right? This is all beans. I'm asking you if there's another layer of guacamole on top, right? And there is not. So this is a simple tissue. Does that make sense? Okay. So this is simple. Can you tell that the cells are taller than they are wide? So it's likely to be columnar. And then what's your second confirmation that it's probably columnar? Nice oval nuclei, which works almost every time, but not every time. Okay. So we're going to go through a couple of different iterations of simple columnar epithelia before we talk about the cilia and microvilli. And then we'll tell you how to tell the difference between those two, okay? So next picture, can you note to self also, if on a test or a quiz I point you to an area to look at that area, because I know that another area is confusing. And I'm never trying to mislead you, I promise you, unless it's a bonus question, and then I do try to mislead you, because it entertains me. It's bonus, right? Let's see what you can do with like half the information and throwing at you a curveball. You're like, let's see what I can do. And you always get perfect credit on those. Okay, but if I point at something, look at that area. Because some areas are undergoing cell division and they're weird looking. So I don't point at that area. So I point at this area, right? Okay. Can you look at this tissue and tell that it's simple and columnar and that it's an epithelium? Simple columnar epithelium. Good? Feeling good about it? Okay. So let's look at a couple of different ones. Okay. This one breaks one of the rules, but the other two it adheres to so well that it doesn't even matter. So look from here to here. Simple? Yeah? Columnar? Epithelium? Yes. And the nuclei are trying to break the rules, but they're a little bit ovalish in some ways, right? So you never, remember, you never want to use a single thing as your identifying characteristic, right? You want to have a couple of different ways to check. So good? Everybody good with this one? Simple columnar epithelium? All right, so let's throw a wrench in it and see if you can identify it from here. This is a picture from your textbook. Again, if I were trying to help you, I would point you to here and not here so you didn't get confused as hell. Yeah? Okay. Can you tell this is simple columnar epithelium? Okay. And then just a couple more, and then we'll go back and figure out how to tell the difference between them. Okay. How about this one? And look at the area where I pointed you to. Simple columnar epithelium. Everybody good? Okay. This is a different type of microscopy, but it shouldn't be that confusing. Yeah? Simple columnar epithelium. Okay. So simple columnar epithelium is usually relatively easy to identify, but we've got two different types, and in order to understand the two different types, we're going to have to go back and learn something. You probably covered it in lab, but let's make sure that you actually understand it. Okay, so if we're talking about simple columnar epithelia, they're going to have some things in common. So I'm going to draw two different situations, A and B. And now we're going to start talking about body locations. So instead of just memorizing a picture, you're going to try to figure out why it would go in one location versus another. Does that make sense? Okay. So the first picture I'm going to draw, they're both going to be simple columnar epithelium. Yeah. But this one's going to have little projections. They're protein projections going off the surface. Okay. Looks kind of like a minion. Yeah. Okay. Those little proteins have mitochondria at the bottom, and they're wiggly. Okay? And they are actually projections off the surface of the cell. Okay? So the second one does not have projections off the surface of the cell. What it has is the apical surface is really, 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 really curved. So instead of looking like a minion, it looks like Bart Simpson. Yeah? And what curving the surface does is increases surface area so you can do more transport, right? If you're trying to bring something across a cell membrane, if you have more cell membrane, can you bring it slower or faster? Faster, right? If you've got more cell membrane, you can do more transport. Okay, so these are called cilia. I'm going to show you some more details about cilia in just a second. The purpose of cilia is to wiggle things across the surface of the cell. Yeah? And these are microvilli. And these are not wiggly. The purpose of microvilli is to increase surface area for transport. Okay. Now I'm going to tell you two different body locations, and I want you to figure out which one goes in which body location. Okay? All right. One of these two tissues, A or B, is found lining what you probably call the fallopian tubes. Call them oviducts or ovarian tubes. Which one? Yeah, A. Yes? One? Move the A. Yeah? And one of these is found lining the lumen of the small intestine. Which one? B. What's the function there? What are you doing in your small intestine? Boatloads of absorbing nutrients, right? Do you want to absorb half of the glucose and amino acids that you eat? No, you want to absorb all of them if you can. Yes? Okay. So this increases surface area for transport. Okay. Now all we have to do is to figure out how to identify each one. Good? Okay. So to me, if you look at microvilli, what it looks like to me is that somebody took a highlighter and highlighted the top of the tissue. It's very, very tidy and even. Okay. Versus cilia, which look like bedhead. Do you see? Yeah? Cilia look like bedhead on the surface. Does that make sense? Okay? All right, so let's look at a couple of different types and then talk about naming. Okay? So when you name these, typically they either call this one non-ciliated simple columnar, or a lot of times they even leave that off. You can put it on if you want to. It's fine. You can call it whatever it calls it in your textbook, but it definitely does not have cilia. So this is simple columnar epithelium. Now, I have something important to tell you about lecture. In lecture, you are responsible for body locations and you're responsible for specific body locations. And by specific body locations, I mean shrink me down magic school bus style and get me to the right location to encounter this tissue. So tell me where I would find this tissue. We just did the example. Be more specific. The small intestine is, yes, and the small intestine is composed of multiple tissues, right? How do you get me magic school bus style? You say lining. Yes? Lining the small intestine. Because what's immediately deep to this, but still small intestine? Connective tissue. And if you want your small intestine to wiggle, it's going to have what deep to that? Smooth muscle, right? Okay. So you have to get me to the specific location, right? So you find this, for instance, lining the small intestine. So that is microvilli. Does this look like microvilli now that you know? Yeah, it does, right? Looks like a relatively tidy highlighter along the surface and not nasty looking bedhead along the surface. Okay. So this one is hard to tell which one it is, so I wouldn't ask you about microvilli or cilia on that one. This one is, of course, cilia. Okay. Give me a body location for this tissue. Lining the fallopian tubes, yes? Later on this semester, you won't be able to call them fallopian tubes anymore, but later on, but that's later, okay? Lining the fallopian tubes, good? Okay, and this is called, you need one extra adjective. This is called ciliated simple columnar epithelium. Yeah? Ciliated simple columnar epithelium, okay? And can you tell that this one is cilia now? Yeah? Ciliated simple columnar epithelium. Everybody good? So it's really not that hard after you sort of know what you're looking for. Let me show you what microvilli actually look like if you use a different type of microscopy. Oh, here it is. This is called transmission electron microscopy. If you look at transmission electron microscopy, look at the surface. That's microvilli. Can you see that it's basically just curved cell membrane on the surface? It's really, really regular. Okay. And then this other cool picture. This is scanning electron microscopy showing you both from the apical surface. Can you see the difference of cilia and microvilli from the apical surface? Yeah, they look dramatically different from the apical surface, right? That makes sense? Okay, so there's two different types of simple columnar epithelium, right? And so if you see cilia, you have to name cilia. Yeah? Good? Okay, everybody good with that? All right, so now we are moving to the stratified tissues. The stratified tissues have anything more than one layer from the apical surface to the basal surface. Okay, so let's do this one first. Okay, so this is stratified squamous epithelium. And again, ask yourself, can you tell that it's stratified? Yeah, more than one layer before you get to the basement membrane. Squamous, remember, refers to the shape of the cells at the apical surface. So do they look squamous at the apical surface? And I agree that they don't once you get down to the bottom. That's why I have to tell you specifically. You're looking at the shape of the cells at one surface. Yeah? Okay. So stratified squamous. And then can you tell it's an epithelium? Yeah, because it's got polarity, right? It's got one free side and one not free side. Everybody good? Okay. There's two different categories of stratified squamous epithelium, as you know, and I'll teach you how to tell the difference between them. So stratified squamous epithelium, before we get into the difference between them, is this tissue going to be fantastic at transport? No, it's going to be relatively protective. That's what it's going to do, right? You're going to have a bunch of layers before you get down, for instance, to nerves and blood vessels and that kind of stuff. So this tissue is called moist or non-keratinized. Either one is fine. Stratified squamous epithelium. And you find it in locations that are subject to abrasion, but also have a source of moisture. So they don't have to worry about waterproofing, but they do have to worry about abrasion. Does that make sense? Okay. So you will find this, for instance, lining the mouth, lining the vagina, lining the distal end of the rectum. Yeah? So abrasion, but they have their own source of moisture. Does that make sense? Okay. So this is moist or non-keratinized stratified squamous epithelium. How do you tell whether it's keratinized or not keratinized? All right, so let's look at the other one, okay? This is called keratinized stratified squamous epithelium. People have trouble memorizing the difference between the two of these. What this is imagine you took your moist, right, and and then you left a big old pile of dead cells filled with waterproofing protein on the surface. None of those are alive. They don't have nuclei. So what you're going to do is you're going to have nuclei from here until you run out of living cells. You're going to have a pile of dead stuff on the top. The way that you tell that it's dead is that you don't see nuclei going all the way to the top. Does that make sense? Okay, so see if you guys can identify this one. It's a little challenging. Is it moist or keratinized? It's moist. Look what I see all the way to the top. Yeah? I see nuclei all the way to the top. That's moist stratified squamous epithelium. Good? Let's see if I think I got another one down here. What about this one? That's keratinized, yeah? Okay. What about this one? Yeah, that's moist or non-keratinized. Does that make sense? Okay. So you just look for nuclei all the way to the top. If you see them all the way to the top, and it doesn't mean like you have to see them in every single part, but if you see nuclei, right? There's one right there. This is going to be moist. Okay, good. Okay, so where you find the keratinized kind, like this, or like the picture they had in your textbook like this, you find this in places that are subject to abrasion and water loss. Yeah? So the keratin is a waterproofing protein that you pile on top, and it keeps you from losing fluids. So tell me a location for this tissue. Be more specific. Epidermis, or if you couldn't come up with the word epidermis, because you may have not learned that yet, you could magic school bus meat to the superficial surface of the skin. Yeah, that would do it as well. And it seems arbitrary, but I'm going to tell you there's other types of tissue in the skin. Yeah, just below that there's different kind of tissue. So skin was fine when you were in 7th grade. But now we need to be a little more specific, and we have the tools to do so, right? Okay, everybody good? Okay, so those are the two types of stratified squamous. Now, stratified cuboidal, these are stratified cuboidal, I think is pretty easy. Stratified cuboidal is just usually 2, sometimes a little more than 2 layers of cells that are about as wide as they are tall. Now, I acknowledge that you can't see the boundaries of the cells here, but can you see the suggestion that they're about as wide as they are tall? And do you see more than one layer of nuclei from the top to the bottom? Yeah, so that is a stratified cuboidal. Again, what kind of section is this? Longitudinal section. Okay, so now let's look at the same tissue, but this time a transverse section. Do you see? Stratified cuboidal epithelium. You find this tissue lining lots of ducts, not quack quack, but the other kind, ducts, right? Ducts you can see right here, right? Duct of a sweat gland, for instance. That's where you're going to find that tissue. Okay, good? All right. I think that one's really easy to identify personally. I think all of the cuboidals are really easy to identify. Okay, and then next is the stratified columnar epithelium. And this one gets a little confusing until you realize that there is an easy way to tell it from the tissue that you would confuse it with, and I will tell you that in just a minute. So if you look at the top picture, do you see at least the suggestion of more than one layer of nuclei? Yeah? And do they look more squamous, cuboidal, or columnar? Columnar. And then look at the shape of the nuclei just for a little extra confirmation. They're oval, right? So this is stratified columnar epithelium. It's probably easiest to confuse with pseudo stratified, but I'll teach you how to tell the difference in just a few minutes, and then you won't screw it up again. Okay? So this is stratified columnar epithelium. It is not found very many places in the body. The only dependable place that I can tell you that it's generally found will be in the distal end of the male urethra. Okay? So about the last 1/3 of the male urethra. Why not female? But the female urethra is only about 1.5 inches long from the urinary bladder to the external urethral orifice. But the male urethra is about 8 inches long. I'm not saying penises are average 8 inches long because I was not born yesterday. But all the way from the inside of the urinary bladder all the way to the tip of the penis with the external urethral orifice is around 8 inches long, and it changes tissue type as it gets to the distal end. I'll tell you about the other tissue in just a minute, okay? And at the distal end, it is stratified columnar epithelium. And it has to do kind of with the double functionality of the male urethra. The female urethra, in a typical anatomical female, serves exclusively urinary functions, yes? in the male urethra, and a typical anatomical male will serve at different times, and there is a protection mechanism, urinary function and ejaculatory function, right? And so there's some tissue specialization that has to do with that, and I'll teach you guys about that later. Okay, so this is stratified columnar epithelium, and this is also stratified columnar epithelium. Do you see? Okay. So we've got stratified columnar epithelium here as well. Everybody good with that? Okay, so where do you find stratified columnar epithelium? Lining the distal end of the male urethra. Yeah? Okay. The other type of stratified epithelium is not called stratified. They just broke the naming convention here, but it is a stratified tissue, and it's called transitional epithelium. Transitional epithelium is a stratified tissue that looks dramatically different when it's stretched versus unstretched. So let me tell you where you find this tissue, and then we'll go back and look at the look of this tissue. You find this tissue lining the vast majority of the urinary tract. So urinary bladder, lining urinary bladder is a great place to think about it, because of course the urinary bladder can be empty, right? Or it can be full, and it can usually contain around a pint of urine when it's full. Okay, so this is what transitional epithelium looks like when the urinary bladder is not stretched, meaning empty. Notice that you can tell that it's an epithelium, yeah? It looks stratified, yeah? But it doesn't look columnar, and it doesn't look cuboidal, and it doesn't look squamous, right? So, and then something characteristic of it when it's not stretched is that the cells at the apical surface look domed. Do you see how they look kind of like domed on the surface? Okay, now, I never test you over this, but what do you think this tissue is going to look like when you stretch it? Yeah, it looks like stratified squamous epithelium. It totally does, which is why I never ask you it stretched, because that's mean and you have enough crap to do, right? So this is the relaxed look of transitional epithelium, and you are going to find this lining the vast majority of the urinary tract, meaning urinary bladder for sure, yeah? Urethra, whole female urethra. In males, that proximal 2/3 was lined with this. And then the ureters, right? The ureters go from the kidneys to the urinary bladder. That's lined with that as well. Good? So that's transitional epithelium. Now see if you can identify it a couple more places. Again, look for a stratified tissue that is domed on the surface and doesn't really fit the other criteria. Ready? Can you tell? Yeah, that's transitional epithelium. How about that one? That's transitional epithelium as well. Everybody good? Okay, so transitional epithelium is a stratified tissue, but it's not called stratified. Good? Okay, and that's the last of the stratified tissues. And now we talk about our friend, the pseudo stratified tissue.
Transitional looks like bowling pins, kind of.
Bowling pins? Oh yeah, it kind of does. That's fun. Yeah, like little happy bowling pins. Okay, so now let's talk about pseudo-stratified and why it's called pseudo-stratified and how you can keep from confusing it with other things. It's not terribly hard once you get used to doing it. So pseudo, of course, means what? False, yeah. So this tissue looks stratified, but it's not, and the reason it looks stratified is because although all of the cells are attached to the basement membrane, they're really different heights. So, ready? One of them's stuck there, one of them's there. This one goes all the way up over here. I got a couple of little ones, right? And this one goes up over here. So can you see that it gives the suggestion that they're stacked, but they're actually all attached to the basement membrane? Yeah? So it's pseudo-stratified. Yeah? There actually is just one layer, okay? So how the hell do you tell this one? Well, you tell this one because it looks stratified, but it's also the only other tissue you're going to learn that has cilia on the apical surface. So the deal is that if you've got cilia, it can only be one of two tissues, yes? You do have more ciliated tissues, but I'm only teaching you two, okay? So it can only be one of two tissues, and that means you only have to be able to tell it from the other ciliated tissue, which makes it way the hell easier, doesn't it? Because can you tell this tissue, look at it real quick, from this tissue? You totally can, right? So if it's got cilia, do you hear how the logic goes in your brain? You narrow the choices from 12 to 2. And then all you have to do is tell it between the two, right? So this is ciliated simple columnar epithelium. And this is pseudostratified, ciliated, columnar epithelium. I don't care which order you get those adjectives in, because nobody agrees on the order. I like all three of them. You can write down what your textbook, I don't think your textbook uses ciliated every time, or maybe it doesn't use columnar every time. Go ahead.
So are you saying that pseudostratified always has cilia on it?
The pseudostratified that you are learning always has cilia on it, and I won't use another for quizzing you. Yeah? There are other tissues, I'm not going to throw those at you. Okay? Yeah? So the pseudostratified always has cilia on it. So now, is it easy or hard for you to identify this tissue? What is this? No, that's not simple. You see the suggestion of multiple layers? Yeah? And cilia? Yeah? So this is ciliated pseudostratified columnar epithelium, right? And now let's look at a couple other questions. What is C? What is the structure that C is pointing to?
Cilia.
Yeah. Okay. What if I said, what is this surface? Apical. What is this surface?
Basal.
Basal. We haven't learned these yet, but those are goblet cells, and we'll talk about them in just a second. Good? So, if you got cilia, there's only two possible answers. Good? Okay. So that's pseudostratified. Now we already did the cell shape, so we can skip past that, and then we're almost ready to show you all of the possible questions you could get from an epithelial tissue. Okay? So the other thing is to make sure that you understand the possible apical surface features, like what could be on the surface. And we already did cilia. We already did microvilli. The only thing that we have to talk about now is goblet cells, which is what we're seeing here. So you can see a goblet cell right there. You can also see them here. Let me show you what you're actually looking at when you see a goblet cell and why they look like that. So what a goblet cell looks like is it's a long, skinny cell, but usually the nucleus is darkly staining and it takes up the bottom. And what it does is it makes it look like a skinny to wide cell, like a goblet or a wine glass, yeah? Okay? And goblet cells' function is to produce a protein called mucin, which when you mix it with water becomes mucus. And they produce it and they dump it onto the surface. Yeah? So let me show you that. So here's a goblet cell up really, really close. But you can see, look, right? The nucleus takes up most of the bottom, so it makes it look skinny. All of these are little vacuoles that are filled with mucin, which I'm going to dump on the surface, and then it'll become mucus. Does that make sense? So that is a goblet cell. And goblet cells are found in tissues that need mucus. So let's go back and look at the tissue that we were just looking at and talk about where this tissue occurs and why you would need mucus here. This tissue has wiggliness and the capacity to produce mucus. Yes? It's got those two things. Okay? This tissue you would find lining the upper portions of the respiratory tract. Okay? So what do the cilia do there? They wave up, yeah? Because you live in Salt Lake or Provo, you are breathing in lots of particulates. You do not want them to get to the respiratory surfaces of your lungs. What happens as long as you are not a smoker, smoking paralyzes the cilia. What happens as long as you are not a smoker is these things are consistently waving up towards your mouth so that you can, and then after you do that, what happens is the mucus actually catches or traps the particulate matter, right? And then you swallow it or spit it out. Nobody wants to know what you do. Just don't make us listen to it. That's all we have to say, right? So they call that the combination in this tissue, ciliated pseudostratified columnar epithelium, which is lining the upper respiratory tract. They call that the mucus elevator. That's sexy, isn't it? So the mucus elevator waves up and catches it, right, in the mucus, so that it doesn't get into the deeper portions of your lungs. That's your mucus elevator. So let me show you how your mucus elevator works. Here's a little video of the mucus elevator. You ready?
One defense mechanism mammals use to prevent invasion by pathogens.
So you can see it's all waving one direction, right? And then we're going to see a top view of the mucus elevator as well.
Sweep mucus and particles into
right?
the throat.
So it's moving the particulate, so the mouth would be up here. That's how that works. So that's a goblet cell. So we run into high numbers of goblet cells, really primarily in two tissues. I mean, there are exceptions of other tissues that you didn't learn. But we know what the mucus is for here, right? It's for capturing those particulates. Now I'm going to go back to a tissue you also learned that has boatloads of goblet cells most of the time, and ask you if you can figure out what this tissue has goblet cells for. Okay. So this tissue right here. First you have to try to see if you can remember what the tissue is and where it's found, and then start figuring out why the hell it has goblet cells. So what's the tissue? Simple columnar epithelium. Where's it found? Lining the small intestines. Now, what do you think the goblet cells are for there? Yeah, they actually are just lubricating. They're just keeping stuff moving through the digestive tract. Does that make sense? Okay, so if you see a whole bunch of goblet cells, you start thinking two tissues, right? Okay, so let's look at this. This is just to sort of narrow it down for you. If you see cilia, you know what they're for, and you only learned two ciliated tissues, so it's got to be one of those two. What are the two ciliated tissues? One more time. Ciliated simple columnar epithelium and pseudo stratified ciliated columnar epithelium. Good, right? If you see goblet cells, in all likelihood, it's one of the two tissues that you just said, which was pseudo stratified ciliated columnar epithelium or non-ciliated simple columnar epithelium, the other one, okay? If you see microvilli, the only tissue that you learned with microvilli was the non-ciliated simple columnar epithelium. Good? Okay, so these end up helping you narrow down your identification if you use them properly. Good? Okay, last little tidbit, and then we'll talk about the study tips. How do you know when you are finished studying an epithelial tissue? You do not do it the way that most students do in lab because they do not do well when it's a question that they didn't expect. So what people end up expecting is, hey, name this tissue. But that's not the questions that I'm always going to ask, or the person in lab is always going to ask. So before we get to that, let's talk just briefly about what we call the lateral cell junctions in your epithelial tissues. So here's what we talked about before. We talked about the fact that epithelial cells tend to be right next to one another, right? And they're all stuck to a basement membrane. So let's use a metaphor to get through with this, okay? So if we are talking about epithelial tissues being stuck to the basement membrane, most of them are on surfaces. So not only do they need to be stuck to the basement membrane, they also need to be stuck to one another so that I have some control over what gets through. Does that make sense? Yeah? And so the metaphor I want to use is epithelial tissue on the top of another tissue is kind of like tiling a floor. So if you're going to tile a floor, and let's make it simple because it works with the metaphor better. If you're going to tile a floor, then the first thing you're going to do is look at, of course, the surface of the stuff that you're tiling. That would be like the surface of the what tissue that's underneath? What's underneath? Connective tissue. Okay. And then the first thing you're going to do when you tile is you're going to put down adhesive, right? And the adhesive is what? The basement membrane, exactly, right? And then we're going to put the cells right next to one another, just like an epithelial tissue. But we're not done, are we? We've still got to put the grout in, yes? And the grout is what we're going to talk about right now. There's 3 different categories of grout in tissues. And what those are based on what you need the tissue to do. So the first one is called a desmosome. And a desmosome, I want you guys to imagine that this is an epithelial tissue, yeah? You guys are the connective tissue underneath. There's a basement membrane right there, okay? Now, the desmosome is about making this tissue really strong, really resistant to stretching and pulling. So what would happen with these 2 cells to make it really strong is she would reach over and grab onto your cytoskeleton and you would reach over and grab onto hers, right? And then they would have a really strong weld between them. So it's basically like 2 hemidesmosomes, yeah? Hemidesmosome is just 1 cell reaching in. This is 2 cells, yeah? Hemi means half, yeah? So this is a desmosome. Now, desmosomes are really strong, but they're not really impermeable. They're really for strength. So what I want you to imagine is now they're holding on to one another and we're going to play a game of Red Rover, right? If I tried to go through a grown adult, they could probably keep me out. Now I'm going to have a toddler and a kitten go through. They can totally get through, right? They can get around the desmosome. So it's not impermeable, yes? But it's really strong, resistant to stretching and pulling. You with me? Okay? Okay. So let's talk about how you would make it impermeable. So now these 2, what they want is they want to protect you. So now I am the stomach acid, which has a pH of about 2. You guys are the epithelial tissue that's lining the stomach, And you are modified so that you can handle that pH, but you guys are the connective tissue cell, and you cannot. So what you guys need to do is a waterproof scuba zipper between the two of you, right? And that is going to make me impermeable. It's not necessarily strong, because I can pull on you this way and yank it apart, but it is impermeable, and you can see it here, right? So this was just a little spot weld. This is a three-dimensional zipper, right? That goes all the way between both of those cells. So it has to zip both of you all the way up, right? Does that make sense? So that's called a tight junction. Tight junctions are about impermeability. Either you don't want to let something out or you don't want to let something in without controlling it, yeah? And then the last one is a gap junction, these guys right here. And gap junctions are about communication, okay? So with a gap junction, what you do is you take two cells and you stick them together. She has a channel protein and she has a channel protein, and you stick them together. And then whatever communication she gets, she will get immediately after. So it allows for wave-like communication between the cells, okay? So it's not strong, it's not impermeable, but they are going to communicate. Okay, so now these guys are going to be my mucus elevator. I know you wanted to be a mucus elevator today, didn't you? Okay, so what I need to tell my mucus elevator is the mouth is up there. Which direction do they need to wave? That direction. So what you need to do, and this is how your epithelium works, is I give her a signal to wave, and then she passes it on. And you guys are going to play, or else everybody's going to get emphysema and die. Okay? No pressure. Ready? Wave. Nope, you're not doing it right. Hands up. There you go. Keep going. All right, who's going to kill us? There you go. Thank you, mucus elevator. You did it. That's how gap junctions work. They make cells work as a single unit and behave together. So, what if a tissue needed both communication and strength? What would you do? Exactly. What if a tissue, like the skin, needed both impermeability to protect from water loss and strength, because you're always yanking on it? Desmosome is a tight junction. So you can mix them up and use different kinds of grout based on the needs of the tissue. Okay, so now, how do you make yourself a successful histology student? Well, you need to realize what could be asked of you. You ready? So this is the study tip. With any histology slide that you run into, you should be able to answer any or all of these questions. So let's just look at this one, okay? So actually, let's look at this one. This one's a little easier for you guys. Okay. So with any histology slide, you should be able to tell me the general category of tissue. And there's only 4 answers that are possible for the general type of tissue. So what is this? It's an epithelium, yes? And then you have to tell me the question that everybody thinks is the only question they're going to get asked, which is name this tissue. And it is simple columnar epithelium. That's as far as most people ever go, and they don't do very well on histology because you are not conceptualizing the other things that I could ask you. I could also ask you, what's this white space? Lumen. Or I could ask you any modified structure that you could see. On this one, I can ask you, what is the structure at the apical surface? Yeah, that is microvilli, right? But sometimes I could ask you, what is this structure? And that is a goblet cell, right? So any modified structure in there is fair game, right? And then I could also ask you, name this surface, yeah? Apical surface, name this surface. Basal surface. I could ask you, name this feature or structure right under here. Basement membrane. I would probably ask you a better one than that because you can't see it very well, but just theoretically. And then here is where you will miss at least 6 questions on your first test if you do not study. For every tissue, you need a specific body location. Now, because I'm not making you memorize all of the specific body locations, that means it's going to be what kind of question? It's going to be a fill in the blank. Yeah? Otherwise, I would have to have you memorize every single body location for every single tissue. Yes? So now, do it for me. Name a specific body location for this tissue. Lining the small intestine. Let's do a few more. You ready? Name a specific body location for this tissue. Lining of the mouth, lining of the vagina. Good. Name a specific body location for this tissue.
Urinary bladder.
Be more specific. Lining the urinary bladder. Exactly. You got it? Okay, so that will be a fill-in-the-blank question, which means that you won't know a damn thing if you haven't studied, right? You're not going to know what to put. You're not going to get it right. And there are at least 6 of them on your first test. They're not all epithelial, but they are all name a specific body location. Good? Okay? All right, so last little tidbit about epithelial tissue. Really, you don't have to memorize slides for this. I just want you to be able to understand this conceptually. Most epithelial tissues that you are going to learn are on surfaces, yeah, inner or outer surfaces, okay? But there's a different category of epithelial tissue that is also epithelial tissue, but isn't always found on surfaces, and it's called glandular epithelium. And glandular epithelium, the histology slides are really hard. That's one of the reasons we don't have you learn it. But I do need you to know it fits into the category of epithelial tissues, especially because it's got this one really important thing in common with other epithelial tissues. It is predisposed to really rapid rates of cell division and regeneration, and keep that in mind, your minds, when you start thinking about clinical applications. So, glands or glandular epithelium is made of epithelial tissue. And all a gland is something that produces and secretes a product. So this is like the simplest gland you could have, right? This is a unicellular gland, right? Okay, that makes sense because it produces and secretes a product. Okay, so now we need to separate glands into exocrine and endocrine. Okay, endocrine glands do not secrete their products onto a surface. They secrete hormones into where hormones go. Where do hormones go? Bloodstream, yes. So you're not, for instance, going to find the pituitary gland near the surface, but it is glandular epithelium, okay? And we don't have you learn the histology slides because they're hard. They're hard to do, but it is glandular epithelium, okay? Now, the other kind of gland is called an exocrine gland. An exocrine gland secretes its product onto a surface. Now, with that in mind, is this an endocrine or an exocrine gland? That's an exocrine gland, okay? Not all exocrine glands are unicellular. Some of them are multicellular. When they're multicellular, you can see them sort of folding. But if you look at it, guys, doesn't that actually look like epithelial tissue anyway? Yeah? So an exocrine gland typically has a duct leading to a surface, right? And sometimes you can see a pore if you're just seeing the top of it. Endocrine glands don't have that because they're not associated with surfaces. Yes? Okay, so these are exocrine glands and endocrine glands. Now, before we go any further, exocrine glands secrete their products onto surfaces. They don't have to be inner or outer surfaces. It could be either one. So I want you guys to brainstorm for me and think of some exocrine glands. Sweat glands, absolutely, yes. Sebaceous and oil glands. Go ahead. The glands associated with hair follicles, yes. What else? Yeah, those are all different kinds of sweat glands. Good. What about, okay, I'll give you guys some glands. You categorize them for me. You ready? Thyroid. Endocrine or exocrine? Endocrine. Salivary. Exocrine. Mammary glands. Exocrine, yeah? Pituitary gland. Endocrine. Lacrimal glands, tear. Exocrine, right? Does that make sense? Okay. Yeah, if it's going to a surface, it's exocrine, right? Why do these end up being clinically important? They end up being clinically important because think about this for just a second. Most of us are going to encounter cancer either in us or in someone that we love because we're long-lived species, and cancer tends to come up in long-lived species. So, what do you think the predisposition to rapid rates of cell division and regeneration does to the likelihood for cancers? Increase or decrease? It increases dramatically, yes. So if you have a tissue, and you do, that is predisposed to rapid rates of cell division and regeneration, and then you just have a slight mutation that changes its ability to control that, then it runs rampant and you get a cancer with that tissue. So does it surprise you at all that the vast majority of cancers originate in epithelial tissues? Now let's think about the cancers that you run into most frequently. Skin, epithelial. Breast, epithelial. Lung, epithelial. Ovarian cancer, it's on the surface. It's an epithelial cancer. Now, I'm not saying that all cancers are epithelial, but most cancers right? A big chunk of the cancers that you can imagine originate with epithelial tissues. So they end up being really clinically important. And just as we're all getting super duper bored with histology, and it's not just you, it's me too, remember how important it is for a pathologist to be able to recognize a normal versus an abnormal epithelial tissue. It ends up being incredibly important for you guys to recognize a normal versus abnormal epithelial tissue. So rapid rates of cell division and regeneration. And then you also need to think about what decisions that means that you should make about how you treat your epithelial tissue. Do you take a tissue that has really rapid rates of cell division and regeneration and expose it to carcinogens and mutagens and then hope for the best? No, dumbass, you sure as hell don't do that. That's stupid, right? Now, do you expose it to UV radiation purposely, which is a known carcinogen and mutagen? No, that's still stupid. Don't do that, right? It's bad for you. It increases your lifetime risk of cancer really dramatically. What about the tanning beds? They want to tell you that that's dramatically better for you, but it's because they're trying to sell you tanning memberships, right? It's not good for your skin. You need just enough to get vitamin D, and with the rest of it, you do not need any more of it because UV radiation is a known mutagen and carcinogen, right? So you need to protect your skin. And those of us who are past our 20s and our 30s and 40s also wish we could go back and redo it, right? And not lay on the roof with baby oil and iodine on, right? And get some wrinkles that you didn't want and some spots that make you look like you're 85. So you're going to regret it anyway. And everybody's hot when they're 20. It's much harder to be hot when you're 40. You just got to hold on to it. Go ahead. Did you have a question? Okay. All right. So endocrine glands, we got them. All right. So now let's move on to connective tissue. Connective tissue is the hardest for students. And it's the hardest because it's really, really variable. Like blood is a connective tissue, and fat is connective tissue, and bone is a connective tissue. Super variable in the way that it looks and the stuff that it has in it, and also no one ever agreed upon a naming convention. So it doesn't have like a naming convention that you can predict. Three-name naming most of the time. So it's not easy for most students. I can make it easier for you, so let's start on that. So it's the most common of the tissues. It's also the most diverse of the tissues. It is really widespread with the exception of the fact that if you are not having a terrible day, it should not be on the outside of the body. Like if you got a bone poking out, you had a bad day. If you got some fat squirting out, you had a bad day, right? So it should not be exposed to the outside of the body. It should be almost everywhere else, between things, strapping things to other things. It is usually really vascular, not always, but usually really vascular. Cartilage isn't, but most things are. And then the other thing that it has that's really dramatically different from epithelial tissue is that epithelial tissues tend to be right next to one another like fence pickets, right? And this tissue tends to have a bunch of crap that you have to identify that's not a cell, and then another cell, yeah? So widely scattered with lots of space between the cells and things to identify between the cells. Okay, so let's talk about how you start with the identification process of a connective tissue. First, connective tissue, you need to work on organization, not just memorization, so let's talk about that. First, why on earth is blood considered a connective tissue and bone considered a connective tissue and fat considered a connective tissue? Because we can all agree that they don't look like they have much in common in adulthood. And the reason that they're all considered connective tissues is because they all originated from the same embryonic connective tissue. And this embryonic connective tissue is called mesenchyme. Embryonic connective tissue. You don't have it anymore, but you still have some of the cells left behind, okay? And all of the tissues that originate from mesenchyme are connective tissues. Ready? So, with the connective tissues, you go extremes. So, sometimes a connective tissue is almost completely fluid, and almost everything between the cells is mostly water. Yeah? And you only learned one fluid connective tissue. What was it? Blood, exactly. So, why is blood considered a connective tissue? Because it originated from mesenchyme, which makes all connective tissues. Does that make sense? Okay? And we'll look at what mesenchyme looks like, although it's not very impressive to look at. Okay? Now, the other extreme is the almost completely solid tissues, which are called the supporting connective tissues. And that includes bone, in which the stuff between the cells is almost complete solid crystals. Yeah? And so it makes a solid tissue. Or cartilage, in which it's a really, really rigid gel between the cells. Does that make sense? Okay. Supporting connective tissues include bone. And you're only learning one bone slide now. We'll learn another one when we get to the bone chapter. And cartilage. And you learn 3 cartilages. Good? Okay. So, and we'll do those later. And then the one that's the most difficult, you have 6 of them to learn. So the one that is in between solid and fluid is kind of like squishy tissue, like fat is squishy and tendons are squishy. Does that make sense? So it's not completely solid, it's not completely fluid, it's somewhere in between. And this is called the connective tissue proper. And it has a further subdivision. Loose and dense. So let me introduce you to loose versus dense. You ready? Okay. So loose versus dense has to do with how much crowding, usually of fibers, there is in the matrix. So let me show you 2. This is tissue A. Okay, keep it in mind for just a second. And this is tissue B. Okay. B is more likely to be what? Loose or dense? It's dense, yeah? It's completely packed in the matrix. And A is loose. Does that make sense? Okay, so that's the loose versus dense. And what we are going to do is we are going to learn 3 loose connective tissues and 3 dense. Okay, so that gives you 3, 6, 9, 10, 11 connective tissues. Going. Good? Okay, and you've already learned them in lab. Let's just make sure that they make sense to you. Okay, so for me, FYI, I am an organization junkie, not in my car, definitely not, but definitely in my knowledge, right? And so you have to keep this information organized because instead of just, for instance, asking you to identify this tissue, I might ask you what category of connective tissue it is, and you have to come up with connective tissue proper. And then I might ask you if it's loose or dense, right? And keeping them organized will allow you to look at what the loose ones have in common and start to figure out what that means about those tissues versus the dense ones, right? So the organization is just as important as the memorization. And in lecture, again, you're not just concerned about the what every single time, you're also concerned about the why, right? What is this telling me? Instead of just rote memorization. Good? Okay, which is why some people who are strict memorizers tend to do better in lab than in lecture. Yeah? Strict memorization does not get you much retention, so I will force you to develop that other part. And please always remember that if it comes to a showdown, lecture is 75%, lab is 25%. Yeah? Okay, so if there's a lecture test versus a lab test up against one another to study for, just do the math. Okay. In addition, please remember that I try to make sure that if we ever have them at similar times, they're over similar materials. So I am working on that for you. Okay, so, but I do every once in a while have people who are like the week before the lab practical, they just don't come to lecture at all. And I'm like, well, that's kind of a foolish endeavor, right? I'm like, all right, so you go ahead and do that. All right, so let's talk about connective tissue functionality a little bit, and then we'll get into the basic organization. Okay, so connective tissue does as varied a thing as blood, bone, and fat do. Like it could do any damn thing except probably be good for transport across surfaces, right? It doesn't like contract like muscle, and it doesn't send electrical signals like nervous tissue, but almost everything else that you could imagine it actually does. So sometimes connective tissue is protective. Like in some places in your body, fat is protective. Anybody, do you know any places that it's protective? We obviously do not need to protect our gluteus maximus as much as we do, but yeah, on the surface of the kidneys, it's really protective. Good, right? In some places, of course, bone is really protective. Yeah, like the cranium. There's not a hell of a lot of muscle attachment going on up here. There's teeny tiny little muscles and not a ton, but this is really protective. The ribs are protective, that kind of stuff. Sometimes it is supporting you. So connective tissue is the reason that you can resist gravity, because you actually have these really strong fibers in connective tissue called collagen that help sort of resist gravity. So bone and cartilage have a lot of collagen in them. And sometimes connective tissue is about strapping something to something else. So a tendon straps what to what? Who knows? Bone to muscle. And a ligament is the same tissue, but it's strapping bone to bone. Exactly. It's the same tissue. Yeah? Okay. Sometimes connective tissue is about storage. Bone marrow stores fat, among other things. And of course, adipose stores fat. Yeah? But sometimes it's not about storage. Sometimes it's about all of the things that blood does. All of them are connective tissue functions, both the immunology function, the gas transport function, all of that kind of stuff, the disease fighting function. And sometimes connective tissues produce other connective tissues, like bone marrow is responsible for producing blood. Okay, so that's not meant to be comprehensive in any stretch of the imagination, but let's just briefly talk about how connective tissues are put together. So this is just like a model connective tissue. So basically with connective tissues, you are going to have the cells, yeah, the fibers, and then the stuff that is not cells or fibers, which is called ground substance. Okay, so let's talk about that briefly. And this is stuff that you did before, the prefixes, roots, and suffixes. Okay, so when you're identifying the cells in a connective tissue, and you do need to identify the cells in connective tissue. A lot of people don't remember that they're supposed to do that. The cells will either end in site, blast, or clast. Okay? If you have a cell that ends in site, it generally means it's a relatively mature cell. So for instance, an osteocyte is a mature what cell? Bone. A chondrocell is a mature cartilage cell. Good. Okay. If you have a cell that ends in blast, it probably means it produces something or is responsible for the development of something. So if you have an osteoblast, it's probably going to make what? Bone. Yeah. But you're going to run into a whole bunch of cells that are called fibroblasts in connective tissue. Fibroblasts probably make what? Fibers of some sort. Yeah. And connective tissue has a lot of fibers. Right? So FYI, if you can't remember what a cell is called in connective tissue, guess fibroblast. Right? It's not always right, but it's a good hedge your bets kind of thing. Okay? So, and then you won't see any in this chapter, but you will see them once we look into tissues a little further when we get into other body systems. You also sometimes have clast cells, and an osteoclast would, for instance, do what? Destroy bone, breaks down bone. And it sounds terrible when you say destroy, but remember when you need to remodel something, you actually need to do destruction before construction. Yeah? So that's what an osteoclast does. Good? Now, there are a whole bunch of other kinds of cells that you could find in connective tissue, and let's just think about this conceptually before you guys go any further. Okay, so we've got like, this is a fibroblast cell, right? And then you can have some other kinds of cells, mesenchymal cells will be in there. But quite often you will see white blood cells actually move into the area. This is a white blood cell called a macrophage. Does it make conceptual sense to you that a connective tissue, especially a connective tissue that's deep to an epithelial tissue, is a really, really good place to stick some sentinels, some guards in? Yeah? Why didn't you stick them in the epithelial tissue? There's no room in there, is there? There's no room between cells and epithelial tissue. There's plenty of room between cells in connective tissue. So the tissue that is just under my skin is going to have a whole bunch of white blood cells that just sort of go roam. What about the tissue that's under my respiratory epithelium? Would you imagine that's a great place to put some white blood cells? The tissue that's under my digestive epithelium that does absorption, that's a fantastic place to put white blood cells. So you'll find blood vessels running through because it's really vascular. You'll find some white blood cells. You'll find fibroblasts. And sometimes if you start getting chubby, you'll also find some fat cells moving in for storage. They're like, hey, you got some room in there. And you've eaten like entirely too much In-N-Out burger. So let's go ahead and like store some stuff in there, right? You got some extra storage space in there. Does that make sense? Okay. I am going to let you guys go a little early today because you're a couple minutes ahead of the other class. So we will stop there for today, and I will see you guys on Monday. No more freaking Mondays off.